Hello, everybody. Good afternoon in the US, good morning, and of course, anytime you're watching this afterwards on YouTube. Welcome to today's RNA Collaborative Seminar, which is going to be held by us in Sydney from the UNSW RNA Institute and the RNA Therapeutics Institute at UMass Chan Medical School. My name is Selene Fernandez Valverde, and I'm a senior lecturer at the UNSW RNA Institute in Sydney, Australia. And it's my pleasure today to present to you two seminars from colleagues at the UNSW RNA Institute. The first one will be by Professor Pall Thordarson, which most of us know as Pally. And Pall is the current director of the UNSW RNA Institute, and he's also former president of the Royal Australian Chemical Institute. And he is the program leader and a champion of RNA production and research within New South Wales and Australia. And he's leading the New South Wales RNA Bioscience Alliance on behalf of the New South Wales Vice Chancellor's Committee. He has an extensive career of research and has interest in nanomedicine and light harvesting materials to supramolecular systems chemistry. And he's particularly interested in helping develop better lipid nanoparticles for RNA delivery. And he has received a number of prizes, including the 2012 Lefebvre Memorial Prize from the Australian Academy of Science for outstanding basic research in chemistry by a scientist under the age of 40. Please, let's welcome Pall.
Thank you, Selene. It's a great honor for me to be here to get an opportunity to talk to the RNA Collaborative about what we are doing here at the UNSW RNA Institute. So yeah, my name is Pat Thordarson. I'm always called Pally. Just sort of before I go into my presentation, for those of you who are not very familiar with us here down in Australia, Australia is a growing country with 27 million people. And what maybe is something special is that we have two very large cities, Melbourne and Sydney, both with 5 million people. So this is why we believe, say in the case of RNA, that we can make a very big global impact. And we have some of the best universities in the world located both here in Sydney and in Melbourne. So that's just a very, very brief background, sort of, well, what are we doing here? So I'm gonna give you a quick overview of some of the what the UNSW RNA Institute is. So unless I see someone screaming, hopefully that is showing the right screen. And so the UNSW RNA Institute was established in 2021. And you will see shortly that it was done somewhat in response to initiatives that I was involved in here with the state government. Australia is organized into states, somewhat like the US, and the state of New South Wales is where Sydney is the capital, and that's the most populous state in Australia. So this institute, like in so many cases, didn't come out of nowhere. The University of New South Wales, UNSW, has strengths in many areas relevant to RNA. But when we came together in 21, what we did, which was somewhat unique, is that we brought together people from across three very different faculties, the Faculty of Science, Engineering, and the Faculty of Medicine and Health. But as I said, it sort of builds on things that we had here. In particular, the Australian Centre for Nanomedicine, which was established here 14 years ago, and I was a foundation member of, our analytical centre, which has facilities for NMR, mass spec, microscopy, and many other things. I'll highlight the microscopy centre because I think in terms of, say, super resolution microscopy, it's one of the best centres in the world and definitely the largest here in this part of the world. The Kirby Institute, you will hear from my colleague here, Chantelle Ahlenstiel, later. She's from the Kirby Institute. This is one of the oldest and most prestigious and largest infectious disease research organization in Australia. The Ramaciotti Centre for Genomics, very large genomic centre here, one of the top 2 or 3 in Australia, and does a lot of work with all the hospitals here in the Sydney region, for instance. And then the Children's Cancer Institute, which is one of our affiliated medical research institutions and has been very prominent in using nanomedicine and was one of the very first one to start to work in siRNA here in Australia more than 15 years ago. So when we came together in 2021, as I say, we sampled a team of now 30 scientists from across these three faculties of science, engineering, and medicine. I'm not going to try to read through everyone's name, just to give you some idea though of the diversity of the team. So within chemistry, science, we come from chemistry, the School of Biotechnology and Biological Sciences, which is UNSW powerhouse in molecular biology, and psychology. And so we got people who work in organic synthesis, nanomaterials, nanoscience, biosensing, RNA biology. In RNA biology, I think one of our key strengths is particularly our interested work in non-coding RNA, role of RNA in the brain, where we also have people working on CRISPR systems, the role of RNA in immune system, and then adjacent to this work on in biomanufacturing. Our engineers, many of them are working on delivery systems based on novel material, be it biological materials, polymers, or inorganic materials. Our biomedical friends work on novel ways of understanding how biomaterials, cells, and nanoparticles and so on go through biological membrane, for instance. One of them also is an expert on stem cell reactors and stem cell manipulation. And in medicine, as I mentioned, the Children's Cancer Institute and the Kirby Institute, so they're the two main medical research institutions affiliated with us in this space. And then in the School of Biomedical Sciences, we have people working on fundamental RNA biology, on malaria, T-cell biology. And we also have a pediatrician who is an expert on rare diseases and has been involved in clinical trials on SMA, for instance. And one particular highlights, and you met Selene already. She's one of our 3 new talent hires. So as part of the initiative, this was a very large investment from our university, $25 million lot. Part of that investment was to attract new talent, and we were very fortunate to get to attract these 3 new talent in the RNA space to UNSW. As I mentioned, it was a large investment. Now, a large chunk of that investment went into establishing what we call the RNA accelerator, and that's sort of our central facility that brings together then the scientists in the RNA biology, medicine, and chemistry research space. So this is a facility that produces preclinical material. So we kind of normally take over from the first initial hit and help people developing their both the scale and the quality and reproducibility of the material further. And we have 20 staff now in this facility. It's been operational fully for 3 years. It's a ISO 9000 certified facility, still the only one in Australia in that space. And we've done probably now over 700 batches for around 200 projects. Some of the unique capabilities that we have particular strength in chemistry, so we do a lot of work on oligonucleotide synthesis and scaling up on analytical capabilities, particularly including the use of mass spec in RNA quality control and research. For the nanoparticle work also, being able to make peptide ligands, and everything is done, as I say, it's certified, and we try to follow international standards like the US Pharmacopoeia. We established for the same training courses for RNA therapeutic production and quality control. We're involved in many large projects. The one that may be particularly of interest to some of you might be is a collaboration with a company in Boston, Tiba Biotech, and our local Department of Primary Industries, New South Wales, even though it's also very populous, and we have Sydney as the capital, is a large agricultural state. It actually is large. If you're from Texas, it's about the same size as Texas. That gives you maybe an idea. So we've been involved in a project where TIPA has developed an animal mRNA vaccine for diseases such as foot and mouth disease, which are biosecurity threat of great interest to our industry. And we are working on a scale up of that process. And we made the first batch of an mRNA animal vaccine here in Australia a year ago for testing. I sort of mentioned there what's going on here in Sydney and the state of New South Wales. So our state government made a $200 million investment into the RNA ecosystem back in 21. And it's sort of twofold investment. One half of it is for a GMP facility that will be commissioned next year. It's out of the ground already. It sits on a campus of another university here in Sydney, Macquarie University, because this is actually a collaboration between 12 universities within the New South Wales region and the Australian Capital Territory region with the state government. But the state government also put a $120 million fund into a 10-year R&D program in collaboration with us in the university sector. So the state government sees this as an opportunity, partly because it's also invested in a viral vector facility to make Sydney a hotspot for RNA medicine and gene medicine in general. So that's sort of what the state government is seeking with this, and we're already seeing evidence of this happening, that we're starting to get attraction of a lot of interest to Sydney and to Australia. Australia is also a very favorable location for clinical trials, so that's kind of goes hand in hand with this. And yeah, the UNSW RNA Institute is essentially a key entry point into this ecosystem that we are developing here in Sydney and in Australia. There's a lot of effort in Australia in RNA. There's also down in Melbourne, and Moderna has made a large investment there too. So yeah, so for those of you who are heading towards clinical development, consider working with us here in Sydney, because we set up this sort of pipeline where you could then access the GMP material here in Sydney too at the Aurora facility once it's ready next year. But our role is more the entry point and the initial scale up. And then we partner with another university here, the University of Technology in Sydney, to do the final sort of large scale. They have a demonstration clean room that can do some of the larger scale work. So in terms of research, as I say, we brought together 30 academics across several disciplines. We have particular interest in RNA interactions. This includes non-coding RNA, so how RNA interacts with proteins and lipids and so on. Delivery is a big strength for us. Sensing, you'll see that in a moment, one example of that. And manufacturing, particularly quality control. In health, in terms of priority areas, it's infectious diseases, and you will hear that in our presentation here from Chantelle. Oncology, particularly childhood cancer, again from the Children's Cancer Institute. Rare and genetic disorders, particularly pediatric. And then we're moving more and more deliberately towards working on neurological conditions. So just two highlights here, sort of very recent from the group. John Marek and his group just published this very interesting review, and I just want to highlight this because sort of highlights our interest. I'm involved in this too. There's several of us involved in pushing the envelope on where what is the role of RNA in neuroscience. So I encourage you, if you're interested in that topic, particularly the role of non-coding RNA in the nervous system. There's a brand new review here in Nature Reviews Neuroscience from John's group. Sort of at the very other end of the scale, I mentioned detection. The group of Professor Justin Gooding, there's a lot of work on liquid biopsy and amplification-free methods. So here's an example of their work of detecting microRNA in liquid biopsies from single spheroids down at the attomolar region. So it's sort of equally comparable to qPCR, but no sample prep really required. So it's a direct RNA detection method. So they're just two very different sort of bookends of the sort of research people are doing here, but it would be impossible to give you a flavor of what all the 30 different groups are doing here in 10 minutes. And the RNA Institute is highly connected, say, within our own institution with the larger Sydney regions. As I say, Sydney has 5 universities in the region and many medical research organizations, and across Australia and internationally. You can see here Brazil, Indonesia, Malaysia, China, Saudi Arabia, Canada, Singapore, and of course the US. Of particular note is our connection to the New Zealand RNA platform. I'm on their steering committee, and we have had a lot of interaction with a large program there with our friends at McGill. So there's now a lot of mutual connection with the McGill University Centre for RNA Science, and we have had some exchanges already there. and then highlight, as I say, our interest in rare disease work on RNA. And there's a large rare disease network here in Sydney, as the Sydney Children's Hospital network actually has the largest cohort of children that need that sort of treatment in the Southern Hemisphere. So that's it from me. Yeah, thank you.
Thanks, Pally, for a very nice presentation. Is there any questions? We haven't got any in the Q&A. If you have questions, please put it in the Q&A. I'm going to ask you a short one before we move on to the next talk. What do you think is the area that's most pressing for RNA therapeutics?
It's a really good question, Selene, and we actually had recently our advisory board meeting. As I say, although I think scientifically a lot of us, I think you included, are interested in the role of RNA in neuroscience. In terms of the pressing issue, at the more translational end, it's still the delivery challenges. And we just had another meeting about that here yesterday. How can we increase selectivity, target particular tissues and cell types? And it's just not actually about being able to say, get to the brain or get to the lung. It's also then reducing the off targets, in particular in the liver. So we're not there. No one is. But as I say, because we have a lot of people from engineering and from chemistry who have been working in delivery for a long time, this is a key focus area for us. And we're very interested in working with other people around the world on these challenges.
Excellent, thank you. With that, we're going to move on to our next talk. So it is my pleasure to introduce Dr. Chantelle Ahlenstiel, who is a senior research fellow at the Kirby Institute at UNSW, where she leads the RNAi silencing group. She did postdoctoral training at the Vaccine Research Center at NIH in the USA, and then the Kirby Institute at UNSW Sydney. In her group, they work on developing RNA therapeutics, specifically short interfering RNA molecules to silence virus infections. And she has developed she and her group have developed a novel RNA therapeutic design pipeline based on their experience with HIV. And they have applied this platform to develop RNA therapeutics to target other acute and chronic virus infections, including SARS-CoV-2, EBV, HPV, and HDLV1. Welcome, Chantelle. Oh, it seems we might have lost Chantelle for some reason. In the meantime, I will remind you
I do see that she's not on any longer.
I see. It could have been.
Well, I think we can, Julia and Hassan, if you're ready to go, we can circle back to Chantelle
Right.
if she's had some.
You'll have to wait for her.
Sure, sounds great. Yeah, let's do that. I'll introduce Hassan, and then we can come back to Chantelle. Hello, everyone. I'm Julia Alterman. I'm an assistant professor at the RNA Therapeutics Institute at UMass Chan Medical School, and it is my absolute pleasure to introduce our next speaker, Dr. Hassan Fakih. Hassan actually grew up in Lebanon and received his BS in pre-medical chemistry from the American University of Beirut. And he pursued his PhD actually at McGill in chemistry and chemical biology in the lab of Dr. Hennady Slayman, designing and optimizing DNA nanocarriers for nucleic acid therapeutics. Hassan then joined the Kurova lab, Anastasia Kurova's lab, as an exchange student in 2020 and later returned as a postdoctoral associate in 2022. Hassan's current research focuses on enhancing the extrahepatic delivery of siRNA therapeutics, and I think that's what he's going to be talking to us about today. So with that, I will turn it over to you, Hassan.
Thank you, Julia. Can you just confirm that you can see my screen and hear me well?
Looks good.
Okay, perfect. Thank you so much for the kind introduction and for the organizers for giving me the opportunity to present today at some of the work that we're doing here at the RNA Therapeutic Institute and specifically in the Kurova lab on advancing extrahepatic siRNA therapies using chemical innovation. And particularly, I'm going to focus today on improving muscle delivery. I believe I don't need to spend a lot of time to this audience introducing small interfering RNAs, or siRNAs for short, which are now a clinically proven, long-lasting therapeutic modality, which basically rely on this universal mechanism of RNA interference, where a double-stranded RNA molecule, once uptaken into the cell, utilizes the RNA-induced silencing complex, which then retains the antisense strand that is specifically and uniquely designed to target an mRNA sequence of interest through sequence hybridization, and then leading to its degradation. So you can imagine this can lead to precise and selective gene silencing of any target of interest. And this has actually been used and developed for 6, and actually I should say now 7 FDA-approved siRNA therapeutics. What's unique about these therapeutics, they actually provide long-lasting therapeutic effect. And here I'm showing you one example where a single injection in patients can give you a durable effect that could last up to a year from a single dose. However, as rosy as it sounds, siRNA actually took about 20 years since its initial discovery to reach the clinic. And the reason for that is we have to think of the challenging path that the siRNA molecule has to take inside the body for it to be effective. RNAs are known to be readily degradable by nucleases. They also can induce an immune response. siRNAs are also too small. They can be quickly cleared by the kidney, and they have short circulation time. And then even if you bypass all of these issues, you still need to facilitate extravasation into the tissue of interest and improve cellular uptake, which siRNAs unfortunately are negatively charged, and it's also difficult for them to do that. So if we distill down all of these challenges together, what we can see is we have challenges of stability and immunogenicity, pharmacokinetics and biodistribution, as well as productive tissue extravasation and cellular uptake. However, as I should say, chemistry innovation almost solves all the problems. As a proud chemist, I always have to give a shout out to all the chemist heroes who are actually working on very difficult problems and providing solutions. And the first chemical innovation that actually happened in the field is in terms of the RNA modifications. This is a field that has been developing new chemical modifications for almost 50 years now, and these are critical and absolutely essential for any RNA translational research. And basically, these RNA modifications is modifying chemically the different components of the RNA molecule, whether at the base, at the backbone, or the sugar modifications. And in fact, all the FDA-approved drugs or FDA siRNA-approved drugs actually employ the phosphorothioate backbone modification, 2'-O-methyl and 2'-fluoro sugar modifications. And these implementing these modifications enhance stability to nucleases both in vitro and in vivo. They improved the potency, and they also lowered immunogenicity. But what's also important, and I want to highlight early on in my presentation, is it's not just about the chemical modifications that you use, but also how do you use them. So I'm just going to show you a very quick, brief example here where 2 different siRNA molecules that were developed against the same target, they have the same sequence, and they also use the same chemical modifications. The only difference between the top and the bottom is how you use these chemical modifications, and the amount and the position of these modifications can actually take a drug that needs 500 milligrams weekly and actually has been halted in phase 3 and transform it into a drug that needs less than 300 milligrams every half a year. So overall, RNA modifications, which is beyond the scope of this presentation, has overall solved the issue of stability and immunogenicity. And what remains as a challenge for us is biodistribution and productive tissue delivery. And this is where the second wave of chemical innovation was also crucial for siRNA therapeutics, where all of the FDA-approved siRNA therapeutics had chemical innovation in terms of their drug delivery. Most famously is the lipid nanoparticle, which was used in the first approved siRNA, followed by also the most famous trigalma conjugate, which allows for very productive and selective delivery to hepatocytes in the liver. However, a common theme you can see in these FDA-approved siRNA therapeutics is they're all liver targeting. And now the next frontier that everybody's focused on between industry and academia is how can we expand effective delivery of siRNAs to different tissues where we want to treat CNS diseases, we want to treat lung, liver, muscle, and skin diseases. and enable the next generation of siRNA therapies. And the way to do this is to always remember that siRNA therapeutics are actually informational drugs. And this is a unique property for these siRNAs, where if you take an siRNA drug that kind of looks like this, you have your siRNA component, a linker, and a delivery ligand. If you take a closer look of these components, you always have fully chemical stabilization and modification of your siRNA. You have your sequence, linker, and ligand. So the sequence is the only component that actually influences the pharmacophore property, which is responsible for your target engagement. You design your sequence to target a specific mRNA, and that's the only target that will be degraded. The rest of these components actually influence what we call the ionophore property, which is responsible and influencing your tissue accumulation, enhancing stability and activity, as well as durability. And this is what our lab is now currently focused on, which is how can we improve via chemical innovation these ionophore properties to enable extrahepatic efficacy of siRNA drugs. A particular focus for our lab for a while now has been how can we improve muscle and heart delivery. And the reason for that is because there are multiple diseases that are impacting both muscle and cardiac health, such as cancer, muscular dystrophies, myocarditis, strokes, and many, many more. A particular target of interest for us was myostatin, which is a very famous target and known TGF-beta member, TGF-beta family member, and it's known for being a negative regulator of muscle growth, meaning the more myostatin you have, the less muscle growth you have. And you can imagine inhibiting myostatin can actually lead to induced muscle growth, which can give you phenotypes that look like this. And besides, getting amazing tasty beef, you can imagine that inhibiting myostatin could be a clinically relevant target for treating muscle wasting disorders where patients are actively losing muscle mass due to the disease that they have. So how do we get siRNAs to the muscle and to the heart? Well, the leading efforts currently in the field are being led by antibody or antibody fragment siRNA conjugates. And this technology is targeting the transferrin receptor, which is abundantly expressed on muscle tissues. And actually, this technology has been shown to be effective in clinic. It's most advanced in the clinic up to phase 2 or phase 3, and they are being dosed at about 4 to 10 mg/kg quarterly. However, this technology can suffer from certain drawbacks, such as being challenging synthetically and having a very complex scale-up. And also, it requires multiple hours of IV infusion, which also could be difficult for the patients. Another class of molecules that can also enable muscle delivery is lipophilic conjugates, which are conjugates such as cholesterol, C16 chains, and multiple carbon chains that can bind circulating lipoproteins and deliver siRNA extrahepatically, including to muscle tissue. And these actually do have certain attractive properties, such as being fully chemically defined. They can be synthesized in an automated fashion in a few hours. And they're also functional not just with IV injection, but also with subcutaneous injection, which only takes a few minutes. And some of these molecules have started to enter clinical development, but they also suffer from certain drawbacks, such as not being selective to a specific tissue, similar to the antibody fragment siRNA, and also not having a well-characterized toxicity profile. In our lab, a very talented postdoctoral fellow, Dr. Annabelle Biscan, when she was a postdoc in our lab, developed this library of lipophilic siRNAs with different properties and looked which one of them would be delivering to muscle the best. And she actually, one conjugate stood out, which is docosanoic acid, showing the highest muscle and heart delivery compared to all the lipid conjugates that we've tested. And based on this data, we were excited to see if we can deliver siRNA in a functional manner. So what we did at the time, we grabbed a myostatin targeting siRNA sequence that was previously identified in literature. We fully chemically modified as our siRNA, which is always critical, as I've mentioned, and we injected mice with this compound. And what we achieved is, as you can see, about 50% silencing of myostatin in gastrocnemius and the quadriceps, as well as in the heart. But if you look closely here, we actually needed to dose a cumulative dose of 120 mg/kg subcutaneously into these animals to get a modest effect of 50%. And we asked the question, how can we improve this to clinically relevant levels? Because 120 mg per kg is a very high dose. So we decided to take a systematic approach to optimize these siRNAs. And to do that, we wanted to optimize the different components of this molecule. So in terms of the conjugate, we're still going to use docosahexaenoic acid, which we know delivers the best out of the lipids we've tested, but we're also now actively investigating other lipid conjugates. In terms of the linker, we're now exploring, we used a DT-DT linker, which also we've shown to improve efficacy of the siRNA. And we started by screening for a new siRNA sequence that could target myostatin to make sure we've identified the most potent siRNA sequence we could achieve. And in order to do that, thanks to Clemens and Katie, we designed 192 unique siRNA sequences that target both the human and the mouse myostatin gene. And out of this screen, we were able to identify 2 lead sequences that have a lower IC50, meaning they have a stronger potency compared to the previously used sequence in our preliminary studies. So after identifying an improved potent siRNA sequence, we turned to improving the backbone and the sugar modification of this molecule, which, if you remember, they can tremendously improve your stability as well as optimize your potency. And I just again want to bring back the slide to highlight that it's not just the chemical modifications that you use, but also a reminder how you use these chemical modifications. So our current compound that we've tested in our preliminary studies actually does use the current chemical modifications that are used in the clinic in the approved drugs. So we started to think what other chemical modifications we can use. And lucky for us, we work with Dr. Kanyamada, who is a brilliant chemist and assistant professor at the RTI, who recently developed this new chemical modification called extended nucleic acid, or XNA for short, where a simple methylene insertion between the phosphate and the nucleoside can tremendously improve your nuclease resistance, folds and folds of improvement compared to the other chemical modifications, which leads to an extended siRNA half-life in circulation, which in turn, and most importantly, improves the siRNA delivery to our tissues of interest, which are the muscle and the heart. So we decided to see if adding XNA modification to our myostatin targeting siRNA can further boost our siRNA potency. So on the top, we have our no XNA modified siRNA, and in the bottom, we incorporated here in green 2 XNA modifications that Ken has previously optimized the position of these XNAs. And to test the efficacy of these molecules, we dosed our mice 2 doses at day 0 and at day 3, and at day 7, we look at myostatin expression in the muscle. So if we look at the quadricep, here in gold is our previously used myostatin sequence without XNA, and we get 50% silencing. When we incorporate XNA, we're now approaching about 70% silencing just by simply incorporating 2 XNAs at the 3' end of the antisense strand. When we combine XNA with our newly identified sequence, we are now approaching 80 or 85% silencing of myostatin, clearly showing that XNA boosts the efficacy of our scaffold. And this is obviously also observed in the gastrocnemius as well as in the heart, really highlighting that using XNA improves the efficacy of the molecule. The second thing we wanted to test is how we use these chemical modifications. So after incorporating XNA, we wanted to explore the pattern of the modification we've been using. So in our original study, we used this pattern 1, we call pattern 1, which has a balanced amount of 2'-O-methyl and 2'-fluoro. And we wanted to change the position of these 2'-O-methyl and 2'-fluoro modifications. So going from pattern 1, we tested 2 additional patterns. Pattern 2 has an alternating modification pattern, and pattern 3 has a 2'-O-methyl rich, so you have more 2'-O-methyl modifications compared to the 2'-fluoro. And the second component we wanted to test is the phosphorothioate modification, which in the top panel, we have 7 phosphorothioate modifications, and then in the lower panel, we decided to drop this modification down to 4. So we took all of these modification patterns, dosed them into the animals, and monitored the myostatin inhibition in the plasma, which allows us to monitor myostatin silencing over time for a period of 1 month. So starting with our original pattern, what you see is a nice 50% reduction at week 1 of the myostatin protein. But as soon as you approach week 2, you start to see this slight rebound of myostatin expression, where the siRNA is starting to lose efficacy or losing durability. However, if you look at pattern 2 and pattern 3, where we simply just changed the positioning and the amount of this chemical modification, now you have a slightly more potent effect at week 1, but most importantly, you see a more durable effect where we do not see any loss of durability of the molecule and no rebound of the myostatin protein expression, at least up to 4 weeks, which is the length of the study, which was observed for both pattern 2 and pattern 3. Now, when we factor in the low PS content versus the high PS content, again, for pattern 1, we see a strong potent effect at week 1, but you start to lose this potency when you approach week 4, whereas for the other 2 patterns, we have a sustained and a durable effect, really highlighting that optimizing the modification pattern indeed improves the durability of our drug candidate. So now that we've optimized the durability by optimizing the sugar modifications and the backbone modifications, we turn towards optimizing the dosing strategy. And that is also an important factor that can contribute to the efficacy of your siRNA molecule. So we asked the question, what is the dosage needed now that we have a more potent siRNA molecule? Do we still need to dose at day 0 and at day 3? And in order to investigate that, we did an ascending dose study by increasing the dose of the siRNA that we are injecting, but we did it in 2 different flavors. The first flavor is by doing a bolus dosing strategy where we just deliver a single dose at day 0, and the loading dose strategy where we do a split dose where we dose at day 0 and at day 3. And then at 2 weeks post-injection, we looked at myostatin expression in the plasma, as well as myostatin mRNA expression in the tissue. So when we look at the myostatin protein in the plasma, what we see first is we have a nice dose-dependent activity, that the higher the dose, the more silencing of the myostatin protein you see, whether it's a single injection or a split injection. However, if you look closely, we also noticed that a 40 mg/kg dose, when delivered in a single injection, is performing slightly better than when it's delivered in a split injection. When we look at the tissue level, again, we see a nice dose-dependent activity. The higher you dose, the more silencing you get in all 3 tissues. But again, we see the same impact. And this was very profound here in the quadriceps, where a single 40 mg/kg dose is performing significantly better than a split injection dose, signaling for us that the single injection could result in a better efficacy in the muscle when delivered as a bolus dose. So now that we've also optimized the dosing strategy for our molecule, we wanted to look at all of the optimized parameters together in order to assess what is the potency of our newly optimized molecules and how durable it is in silencing myostatin in the muscle. So we took our optimized pattern, our optimized sequence, our conjugate, our optimized modifications, and we did a single dose of 40 mg/kg and monitored the myostatin expression in the plasma over time. And now what we see is a single dose of 40 mg/kg actually gives you maximum potency of about 80% reduction that lasts up to 8 weeks from a single dose. And actually, this single dose still gives you significant silencing up to 14 weeks or about 3 months post-injection, which we were very excited about compared to our previous data. But then we asked the question, how far can we push this? How much can we silence myostatin with this DCA siRNA scaffold? And also we were interested in the safety and the tolerability of this muscle delivering scaffold. So we decided to go a little bit crazy and do a biweekly dose of 40 mg/kg. And what this resulted in is almost 95% inhibition of myostatin that was consistent throughout the study, showing you really that the power of chemical optimization of your siRNA candidate can really enhance potency and extend the durability of your effect. But what about phenotypic changes? As I previously mentioned, that knocking down myostatin should induce muscle growth. And in this case, in our study, we monitored body weight, muscle mass, strength, as well as muscle volume. And what was unique about this experiment is because we have the single dose group and the multi-dose group, we're able to compare the effect of temporal silencing of myostatin to a sustained silencing of myostatin and see if there's any phenotypic difference between these 2 groups. So when we first look at body weight, we see a nice significant increase of body weight in both treated groups, whether it's a single dose or a multi-dose group. And we don't see actually a difference between these groups in terms of the body weight. In order to make sure that the increase in body weight is actually a result of increase in muscle mass, we looked at the increase in the lean mass at 26 weeks. And again, both treated groups were increasing our lean mass significantly compared to our control group. But again, we do not see any difference between the treatment groups. And then to confirm that actually the muscle gain is actually functional, we did a grip strength test. And again, we see about 30 to 35% increase in both treatment groups compared to the control, but there is no difference between temporal silencing of myostatin versus sustained silencing of myostatin. And then we took a closer look by doing MRI imaging of the legs of these mice before injection and after 6 weeks post-injection. And what you can see, if you look at the quad and the calf of these treated groups, we see a significant increase of the quadricep and the calf in both groups that were treated with the myostatin siRNA, whereas the control group did not show any significant increase. And when we go ahead and actually quantify the muscle volume from these MRI images, what we see is that both treatment groups resulted in about 50% increase in the quadricep volume compared to the control group. And the similar effect is observed in the calf, which also increased the muscle volume by about 35 to 40%. But yet again, I just want to highlight here that we see a similar effect in this muscle volume increase between the temporal silencing of myostatin and the sustained silencing of myostatin. Specifically, I want to highlight the 19 weeks data where we know for sure that myostatin expression actually has recovered in the 40 mg per kg group, yet we still see that we have maintained that 50% increase in muscle, suggesting that phenotypic benefit does not necessarily need to have myostatin suppressed all the time. And then what about safety? And this is actually also a very exciting outcome for us, because the multi-dose group, which was receiving biweekly injections, actually received a cumulative dose of 520 mg per kg of DCA siRNA, which is an over-exaggerated dose. However, we didn't see any adverse events in these mice. And when we look at some of the toxicity markers we can see in the blood or the CBC readouts, we do not see any change in liver and kidney damage markers, no change in platelet counts, and no change in inflammation markers, really supporting the safety profile of these molecules. And then also there's always a concern in terms of heart damage when you're silencing myostatin. And here we measure troponin and CK-MB, two very well-known heart damage markers. And also we do not see any changes in these biomarkers in any of the groups that we've tested, again, supporting the safety profile of this class of compounds. And then after this data, we asked the question, what is the dosing level that is actually needed for phenotypic benefit? We know that a single dose gives you about 80% silencing that lasts up to 8 weeks. And we also know that biweekly dosing is obviously an overkill because it lasts and gives you more than 95% inhibition for more than 6 months. So what is really the optimal dose to achieve, phenotypic benefit, which is ultimately what we need in a preclinical candidate? So we decided to do a dosing regimen study where we did a 40 mg per kg dose every 8 weeks to maintain maximum silencing. And then we did a 20 mg per kg dosing every 4 weeks and a 10 mg per kg dosing every 2 weeks. And we did this over a dosing phase of 6 months or 24 weeks, where at the 24-week mark, all the mice will be receiving a similar amount or equal amount of siRNA throughout the study. And then at 24 weeks, we stop the dosing, and then we have a recovery phase to see how fast this myostatin expression actually recovers. And throughout the study, we were looking at myostatin expression as well as lean mass and body weight. So to start with the 10 mg per kg group, what we see is actually it takes about 3 doses to achieve about 75% silencing of the target. However, even if you keep dosing, we don't see actually any improvement to the silencing level. We actually reach a steady state of about 75%. When we look at the 40 mg per kg group, what we see is a single dose immediately pushes you to about 75% silencing. And then as you go and dose more and more, we can actually push the silencing beyond 75%, approaching 80 or sometimes 85% silencing. Now, in terms of the recovery, what you can see is that the 10 mg per kg group, as soon as you stop dosing, it starts to recover. Whereas for the 20 mg per kg group, you see that you have a few weeks before myostatin expression to start to recover. Now, for the 40 mg per kg group, again, from the first dose you inject, you immediately achieve 80% silencing, which is what we observed before. And then again, at around the 8 weeks mark, you see that sharp recovery of myostatin expression. However, when you dose again, you push back that silencing down to about 80 or sometimes approaching 90%. And what's interesting is when you dose the third dose, or at 16 weeks, you see that the recovery of myostatin is actually less than that observed at 8 weeks, suggesting an additive effect. So what is the conclusion from here? The conclusion is if you really want to reach beyond 80% myostatin inhibition, you need at least 20 mg per kg every 4 weeks or 40 mg per kg every 8 weeks. The 10 mg per kg only reaches about 75% silencing. And why is this important? Well, it's important because when we look at the phenotypic effect, we see that over the course of the study, only the 20 mg per kg and the 40 mg per kg cohorts reached significant increase in body weight, which translated into a significant increase in lean mass as well over the course of the study, which suggested to us that you really need to hit 80% or more of myostatin inhibition for you to reach a cumulative sustained phenotypic benefit over the course of the lifetime of the mouse. So where does this leave us? In terms of the DCA technology, we know that in rodents, we need about 30 to 40 mg per kg every 6 to 8 weeks. We did not see any observed adverse events, even at exaggerated doses. And we expect in the clinic, this will translate potentially to 8 to 10 mg per kg dose every 3 to 4 months. That can be delivered subcutaneously. However, it's always important to highlight that DCA siRNA will deliver to other tissues, but you can build selectivity of this molecule by targeting targets that are selectively expressed in the muscle. And in fact, Comanche Biopharma, which is a spin-off company from our lab, has completed a phase 1 safety study using a similar compound that relies on a DCA conjugate class, and they have reported safe and well-tolerated doses up to 5 mg per kg. Really encouraging for this platform. So the summary of my work, I hope I was able to show you that through chemical optimization, we were able to improve the potency and the durability of DCA siRNA in the muscle. We can achieve robust myostatin silencing, resulting in phenotypic benefit. And we can actually also highlight here that permanent myostatin inhibition is not necessary to get phenotypic benefit. However, what is necessary is potent silencing. And the next steps for this project is to assess beneficial outcome of myostatin inhibition, hopefully in disease models, and also start to expanding beyond myostatin to other muscle-related targets, which we also started exploring as I show you here. And with that, I would like to thank all the members of the Korova lab, the Yamada lab, and the Alterman lab. It takes a village to conduct all of this work, and I'm happy to take your questions.
Thank you so much, Hassan. Great talk. So we do have maybe time for 1 or 2 questions. If you'd like to quickly type those into the Q&A, maybe I will start. So Hassan, people have tried to go after myostatin in the past without much success. How do you think that this technology compares, and what do you hope to see differently in the outcomes?
Great questions. That is true. Myostatin has been a very well sought out target. I think the differentiating factor for us is all the myostatin therapeutics that have entered the clinic are all based on antibodies or biologics, which are targeting myostatin protein in circulation. We think and we hope that targeting myostatin expression intracellularly at the mRNA level, which will potentially yield a more potent effect and a more durable effect, which hopefully will translate into success in the future.
Great. And we do have one question from an anonymous attendee. It says, great talk. And they'd like to know, is there any system that delivers to the prostate or prostate cancer or other cancers?
Great question. That is another project of mine. We are actually working in the Korova lab as well to improve siRNA delivery to tumors and cancers. I believe we have recently published on another conjugate that can deliver to solid tumors, and I encourage you to look into that. However, I will say we are at a stage where we can reach functional delivery to tumors and cancers, but we don't think it's ideal. There's still a lot of work to improve on that.
Thank you so much. I think I will turn it back over to Selene, but if there are any more questions for Hassan, feel free to type them into the question and answer box, and he can answer them via typing. Thank you so much.
Thank you, Julia and Hassan. Very nice talk. We are now moving on to our talk from Dr. Chantelle Ahlenstiel, who is a senior fellow at the Kirby Institute, and she works in developing antiviral RNA therapeutics. Please go ahead, Chantelle. Welcome. We can see your slides, but we cannot hear you.
There we go. Is that better?
Yes, we can hear you and see you.
Amazing.
Yeah, your slides are not
There we
in
go.
presentation mode. There we go.
Beautiful. All right, so we like to divide RNA therapeutics up into two different classes, one that's dependent on the immune system, so such as all the mRNA vaccines or self-amplifying mRNA vaccines. But the ones that we're interested in are actually independent of the immune system. So I'm going to talk today about short interfering siRNAs. There's also antisense oligonucleotides and microRNAs. And I guess the most well-known short interfering siRNA was the very first one, the Onpattro, that was delivered in a lipid nanoparticle. So thanks, Hassan, for introducing RNA interference. So I'm going to talk more about RNA interference, but I'm going to talk about one of the novel pathways that's really not discussed very much at all. So RNAi actually has two pathways. So Hassan mentioned the post-transcriptional gene silencing pathway, where the siRNA is complementary to the messenger RNA, and it actually ends up degrading it through the AGO complex. What I'm going to talk about is transcriptional gene silencing. So it's a really novel RNAi pathway where the siRNAs are complementary to an actual gene promoter region. And it doesn't end up in degrading anything. It actually ends up in causing chromatin compaction of the promoter region with recruitment of epigenetic marks, which causes epigenetic silencing. And this is actually heritable to the next round of cells. And we call this the block and lock approach because you're actually blocking transcription and you're locking it in this silent state with the repressive epigenetic marks. And so just to drill into this a little bit more, on the left-hand side in the figure on the right, you can see the post-transcriptional gene silencing pathway, which recruits the RITS complex and the AGO2, resulting in mRNA degradation. But what I really want to draw your attention to is the pathway on the right-hand side, which you can either do with non-viral delivery with nanoparticles or with viral vectors, which I'll show you a little bit today. But this exclusively happens in the nucleus where the promoter is located for a gene. And you can do this with short interfering siRNAs or short hairpin RNAs, which I'll show you a little bit today. You can see by the number of citations that PTGS has more than 20,000 from the original Fire and Mello paper, compared to Kevin Morris's paper in 2014 when he first discovered this process in humans. So you can see very clearly which one is more predominantly used. All right, so both pathways have their advantages and disadvantages. For PTGS, we like to use this approach for acute viral infections. So typically in a non-modified siRNA, maximal silencing is approximately 4 days. You can modify it, as Hassan showed, and get that extended silencing for a much longer time. With transcriptional gene silencing, a single dose of siRNA can actually silence out to 3 weeks, totally non-modified. So again, you can imagine what sort of effect we can get once you start to modify these molecules. And so for transcriptional gene silencing inducing siRNAs, we like to use these for our chronic viral infections where you need to get much more long-lived protection. And so this is just a pipeline of some of the siRNAs that we're developing in the laboratory. We've focused primarily on HIV and now SARS-CoV-2, but we've now expanded out to a whole range of acute and chronic virus infections. A lot of them use lipid nanoparticles. I'm not going to talk about today, but we've got a beautiful project with COVID where we've designed an optimized lipid nanoparticle that we can deliver using an intranasal device to the nasopharynx region of the upper respiratory tract. And we're actually going to be adapting this to lots of the other viral infections that are also specific to this region. But today what I'm going to focus on is our HIV project, specifically with a lentiviral vector delivery. All right, and this is for an ex vivo stem cell therapy. As I mentioned, we're also using lipid nanoparticles to do an in vivo stem cell therapy as well. And all of this work was in collaboration with the Walter and Eliza Hall Institute in Melbourne with Professor Mark Pellegrini. And the reason we paired up with them is because they've developed a beautiful humanized mouse model of chronic HIV infection, which I'll walk you through in a minute. So some of the cure challenges for HIV to bear in mind during this presentation is that HIV has a latent reservoir, which is established very early during infection, primarily in primary CD4 T cells. And so these CD4 T cells are located all throughout the body. So the reservoir is located in multiple organs, also sanctuary sites in the brain, and a whole host of different cell types shown here down on the right-hand side. One of the other things that, that siRNA and RNA therapeutics are really geared towards treating is the diverse global subtypes. So while siRNAs are highly specific, the way we design our siRNAs, we do a conservation sequence analysis to start with. And so we choose very highly conserved regions in the virus that are highly unlikely to change. And based on that, and then when you combine multiple ones of these together, we can cover the diversity of HIV subtypes that are out there globally. All right, so the therapeutic I'm going to tell you about today, it's actually a dual therapeutic. So we're using a host factor, which is CCR5. It also happens to be the HIV co-receptor. And so when you knock out CCR5, you prevent virus entry. And this RNAi pathway works through post-transcriptional gene silencing. So it's going to degrade the CCR5 messenger RNA. We're also targeting a viral host factor, the HIV promoter. And the HIV promoter is also called the 5' LTR. And the specific region in the HIV promoter we're targeting is 2 tandem NF-kappa B target sites. And so this shRNA is called SH or siProm-A. And as I mentioned, it induces epigenetic silencing. And so this is working through the block and lock approach. And on the right-hand side, you can see when they're untreated, the CCR5 co-receptor is there, the virus can enter and you get active HIV transcription. On the right-hand side, when they're treated with both the shRNA to CCR5, you degrade the co-receptor. And then when it's also got the transcriptional gene silencing shRNA, you cause that closed chromatin compaction. And so you block HIV transcription. And so it's really in a super latent virus state. So we're basically aiming to mimic what the virus does naturally. All right, and this is just some background data showing how specific the sequences are. So that's one of the great advantages of siRNAs is that they're highly sequence specific. Particularly for TGS, one mismatch will actually knock out the effect, whereas for PTGS, there's some tolerance for mismatches. You can see on the right-hand side in the red is an siRNA, so unmodified siRNA, a single dose, and measuring RT, reverse transcriptase, an HIV gene. You can see that there's silencing out to 15 days with a single dose of siRNA. And this is compared to all the controls, which don't suppress the virus. So just to show that this is actually transcriptional gene silencing and that there are repressive epigenetic marks being recruited following treatment of these siRNAs. Again, the Prom-A is shown in red, and another novel siRNA 143 or a modified version 143T. You can see that there's recruitment of histone 3 lysine 27 trimethylation, which is a classic mark of repressive epigenetic silencing. Histone 3K9 trimethylation shown in panel C is also recruited. And then obviously you expect that AGO1 is also going to be recruited because it's an essential component of the human RITS complex. And then we also expect that acetylation is going to decrease. And then you can see that in panel E, that all of the SI-treated promoter-targeted siRNAs induce a decrease of global acetylation. Okay, so moving on to the humanized mouse model of chronic HIV. So along the top is the actual model. So we start with neonatal mice. They're irradiated to create a niche for CD34 stem cells to be engrafted. for the actual model, the CD34 stem cells are engrafted, and then at about 17 weeks post-engraftment, there's a flow cytometry analysis to determine how successful the engraftment's been. At 17 weeks, you can see engraftment and development of all human lineages of the immune system. So they've got CD4 T cells, CD8 T cells, B cells, all lineages of the human immune system. So that's why we call them humanized mice. So for our model, what we did was once the mice have been transplanted, they're actually transplanted with gene-modified CD34 stem cells, so primary CD34 stem cells. And we had 4 different groups in this experiment. So we had a mock transduced CD34 stem cell, and then we had 3 different groups that had a lentiviral GFP reporter, so we could see what the transfection efficiency was. And the 3 different groups we had was an shRNA that was empty, so it basically just had the loop sequence of an shRNA, and a short hairpin RNA is basically the sense strand with a loop connecting it to the antisense strand. When it's processed and mature, that loop is cleaved and you're left with the active mature siRNA. The second group we had was an SH Prom A group, which targets the promoter. And a third group was actually that dual therapeutic that I mentioned, so the combination of the SH Prom A and the SH CCR5. So we had a transduction efficiency of roughly 40 to 70% in our CD34 stem cells before they were engrafted. And that's a really important point, because while 40 to 70% is quite good for transducing primary CD34 stem cells, it also means that at the flip side, that there's still 30 to 60% of CD34 stem cells that are not gene modified and that are totally susceptible to HIV. And you'll see how that's relevant in some of the data that I show you. So going back up to the top model, at 18 weeks post-engraftment and gene modification, the mice are challenged with HIV, and this is an R5 neurotropic JRCSF strain. They're infected with the virus for 10 weeks, and then they undergo ART treatment. So this is a standard antiretroviral therapeutic treatment. And while ART has been very, very successful in humans, it's actually not a cure. It suppresses and keeps the virus in a latent state, but at any stage, and as soon as you go off ART, the virus can actively rebound to levels of pre-therapy. So that's what we mimicked here. We did an 8-week treatment of ART for the mice. We then did a treatment interruption after 8 weeks of ART when all the mice were suppressed, and then to look at what happens. Does the siRNA therapeutic control rebound or not? So the mice were culled at 40 weeks, and then we measured a whole heap of different parameters. So just finishing up, we can see that the CD34 stem cells, so the CD4 T cells, the humanized CD4 T cells that were gene modified with Prom A are quite stable throughout the time course, particularly at the end at the rebound, compared to the other control groups. And we also did see viral rebound, a delay of viral rebound. So remember how I said that there was still quite a lot of CD34 stem cells that were susceptible to HIV? That's what we found. So there was still quite a few infection that was present, but the Prom A and the CCR5 dual treated groups, as well as our individual Prom A groups, did actually show a delayed virus rebound. We also did some beautiful RNA scope to see what's happening in the tissue, because all the data that I just showed you was in the blood. And what we found in the tissue was that looking at the top 2 panels, the empty control and the mock mice, you can see that there's quite a lot of green virus that was detected by RNA scope. But if you look at the dual treated mouse down the bottom, the representative mouse, you can see that there's actually much less, and that's quantitated on the graphs on the right-hand side. So we can see that the RNA scope data in the tissue, the gene-modified cells are actually moving into the lymph nodes and showing reduction of virus in the tissue itself, which is really exciting. And then we can also see that this was happening in CD4 T cells with the immunostaining. So there we go. Just to summarize and wrap up, this is a really novel RNAi approach, the block and lock approach, and we feel that it's actually quite realistic for achieving an HIV functional cure. We did see delayed viral rebound, and I didn't have time to show you, but we've also measured every single HIV gene that's controlled by that one LTR promoter and found suppression of genome-wide HIV. And we've also done the chip assay to confirm that the epigenetic marks are present as well. And we'd like to now focus on antibody targeting of particularly lipid nanoparticles, but also nanocapsules. And we're also using our optimized LNPs to facilitate nose to brain delivery for the neuropathies that are associated with HIV. And we also think that, like antiretroviral therapy, any HIV cure will likely be a combination approach, whether that's a combination of RNA therapeutics or other cure approaches as well. And with that, I'd just like to wrap up and thank everyone that was involved in this study, in my silencing group, Scott Ledger, Vera Klim, and Anif Hanif, and from the Walter and Eliza Hall, Cody, and from the VRC, Julia. Thanks very much. Happy to take any questions.
Thank you, Chantelle. We are a bit on time, but if there is one question, one person question in the chat, in the Q&A, please go ahead. And if not, I want to thank you, Chantelle, and I'll pass it on to Julia to introduce the next seminar series. I've got a police on behind me, apologies.
Thank you so much, Selene. Okay, well, so our next RNA collaborative seminar series talk is going to be on October 8th, 10 a.m. Eastern Standard Time. And this is actually going to be our first industry-hosted seminar. So please join us to hear from Moderna and Mina Therapeutics. And with that, thank you all again for joining us. We had a great time with you today, and we look forward to seeing you in a couple of weeks.